welcome to what is our 18th Ivan round meeting. And this little thing floating around in front of me is the Ivan Founders Globe that we gave out at the ENC meeting, which is really neat. Why don't we get started with John?
Thank you once again, and thank you all for joining us today, whether you're brand new to the Ivan meetings, a newcomer, or seasoned veteran at this point. As Dave mentioned, this is meeting number 18, workshop number 9, but a total of 18 meetings so far. And gee, I guess we're becoming and continuing to be quite popular. And once again, we certainly are very glad to have everyone with us today. MR Resources and Q1 Instruments bring these meetings to you, of course, and very lucky to be the sponsor of these meetings. MR Resources, of course, been around for 35 years now and providing services, quench recoveries, moves, certainly reconditioned NMR spectrometers, and they're always on the lookout to purchase any unused and or unneeded NMR systems that you may have. I'll ask Don Bouchard to give maybe just a couple of words on Q1, and after that, we'll get things kicked off. Don, if you would, please.
Okay, thank you very much, John. We invite you to get to know Q1. Q1 designs and manufactures complete spectrometers from 400 to 600 megahertz with features for routine use in the research laboratory. Want to upgrade an older system? Q1 can retrofit AS and UltraShield magnets with complete upgrades, including automation, for less than you think. Q1 offers NMR instruments with excellent performance and at an unbeatable price. Q1's operating environment is Spin Studio J. Spin Studio J has a plug-in based paradigm for extending virtually any capability, either internal to the spectrometer or externally via Java-based tools for data acquisition, analysis, and connectivity. Spin Studio J is a multi-platform, multi-language system with a hierarchical data format with validation and exportable to most third-party processing platforms. Recently released is a new NUS plugin based on deep learning algorithm published by Xiamen University and exclusively licensed by Q1. This advanced process uses an iterative process of removing artifacts by a dense convolutional neural network for data consistency to speed data acquisition and processing by a factor of 5 to 10, with improved resolution compared to conventional processing. Want to know more about our consoles and complete systems? Please contact us for a no-risk remote demonstration. Thank you.
Thank you, Don, and certainly a very solid and very capable line of NMR instrumentation, and we ask everyone to look further into these. In any case, I'll ask Krish to give a couple of words on upcoming meetings, and I know we've got a full agenda, but Krish, if you could give us the rundown, please.
Thank you, John. I won't take too much time. I want to introduce the next few topics that are coming down the pike. Just to alert you before we get started with today's topic. The topic for the month of May is QNMR, practical applications. It will be led by Jose Napolitano at Genentech. It's on May 20th. The panelists would be Charlotte Corbett at DEA, David Foley at Pfizer, and Patrick Giroudot at the University of Nantes. We have the June topic is Pure Shift NMR, and the July topic is diffusion and DOSI. The Pure Shift NMR will be led by Matthias Nielsen at the University of Manchester, and the DOSI will be led by Brian Antelek, Bosch and Lomb. We do have a couple of more coming down the pike. Before I give it back to John, I want to invite people to let me know if you have interesting topics that you want us to cover, and even potential panel leaders for that. And we will schedule them as we go. Thank you. Thank you, John. Back to you.
Thank you, Krish. And wow, certainly an awful lot of stuff coming up. And we are, well, look for us to continue these meetings for quite some time to come. That being said, David, if you would kick things off for us today.
Okay, well, basically, I think we have 3 very good talks lined up with our leader, Frank. And as usual, we'll go to roughly the top of the hour with talks and questions through the chat. And then after the talks are over, we'll open it up to talk as long as we want. And it looks like we might have some good discussions today. So with that, I'll give everything to Frank and we can get started.
Thanks so much. Awesome opportunity to address everyone. And it's delightful to see how many people joined in today. Today, our panelists are going to talk about principal component analysis. And we're going to talk about different applications of principal component analysis. In my particular case, I'm going to refer to some work that we do in our groups at NIST, where we are interested in characterizing biopharmaceuticals by NMR. And as an example, this multi-laboratory study that was led by our investigator, Rob Brinson, which analyzed the ability for people to make consistent measurements in this way on monoclonal antibodies. And this particular study generated 375 spectra on 39 different NMR instruments, and we had to analyze it. So Rob is a hero, and he took these 375 spectra and did peak detection of all of them, picked out about 50 or 100 reference peaks, and tabulated the chemical shifts of all of these spectra. You could imagine that it took more than a day to do. All right. But when you analyze a spectrum like this and use the method principal component analysis that we're going to talk about today, well, then you can get beautiful results like this, which can cluster spectra into particular classes, in this case, based just on differences of temperature of acquisition, for example. Now, in this particular study, there were 2 samples that have more than 96% sequence identity. And we wanted to see if we could do different kinds of classification with this data based on the shape of the spectrum, rather than this laborious peak analysis approach that we just showed an example of. And lo and behold, we can use principal component analysis directly on these spectra to do useful kinds of classification. So today I'm going to explain how principal component analysis works and show some examples of how it can extract useful information from spectral series. Okay. In the approach that we're going to talk about, each spectrum in a series is represented as an object in multidimensional space. And so one way of understanding principal component analysis is on the basis of a big multidimensional space, which represents all the spectral data. And in this multidimensional space, we will have one object for each spectrum in the series, and the coordinates of that object are all of the intensities in the spectrum. So if you have a 1D spectrum that has 1,000 points, you can represent that spectrum exactly as a single object in a 1,000-dimensional space. And if you have a 2-dimensional spectrum that has 1,000,000 points, you can represent that spectrum exactly in the same way as an object in a 1,000,000-dimensional space, where the coordinates are simply all of the 1,000,000 intensities in each spectrum. Now, this seems like a bizarre way to organize data, but if we think about this large high-dimensional space, we will see that it has interesting properties. So think about 2 spectra that are more or less identical to each other. Two spectra that are very similar will have similar intensities. And that means in this high dimensional space, they will have similar coordinates. And so in this high dimensional space, similar spectra will tend to cluster. But think of a case, for example, where two spectra are largely similar, but some feature like the intensity or position of one peak changes in one spectrum to compare to the next. Spectra like these are going to tend to lie along lines and curves in this high dimensional space because they will have one set of coordinates in common, all the parts of the spectra that are the same, and some small subset of coordinates that differ in a continuous way. So this big multidimensional space groups together spectra according to their similarities and differences. And what principal component analysis does is in this big multidimensional space, it fits that multidimensional data to a multidimensional straight line, which gives us the direction of maximum variance. And we can project data down along that straight line to generate smaller dimensional scatter plots that reveal the clusters and patterns in this high dimensional space. So this is one way to understand PCA as a kind of projection method from a higher dimensional representation of the data to a lower dimensional representation of the data. So one thing that we do to look at the results of principal component analysis is to look at these kinds of scatter plots. But this principal component vector that fits the spectral data can also be interpreted as a synthetic spectrum. And so another way we can view PCA results are what we call the loading spectra, the synthetic PCA spectrum that represents this principal component. So we have two ways to look at spectral data. For example, we have this collection of spectra from the 375 data sets that we talked about. And these are all kinds of spectra, NUS, conventional, high field, moderate field, and two kinds of samples. So you can spend a month doing peak detection on all 375 spectra, or you could stick the whole bunch into PCA and separate the two kinds of samples very quickly like this. So we're going to talk about how that stuff works. But first, a quick survey of some other applications. And basically, any circumstance where you have a collection of related spectra, you can use PCA to get a nice overview. So, for example, this is a collection of about 200 HSQC spectra mixed with different potential ligands. So this is a ligand screening study. And if we do PCA on the collection of spectra, we can find a particular cluster, for example, where certain peaks exhibit a systematic shift and new peaks appear in the spectrum. So this cluster of spectra here correspond to peaks that have ligands that all bind to the target protein, and they bind with the same binding mode. So information that we can get quickly from PCA. Here's a titration, a ligand titration series, so relatively small number of spectra. We can stick this into PCA and get a PCA scatter plot. And the nice thing about this is, if we expect continuous behavior in the spectral series, the PCA results should also reveal continuous relationships. And there are different ways that we can exploit this too. Interestingly, in other kinds of applications, for instance, a 2D relaxation series, which you would not normally think of as applying principal component analysis to, if we apply principal component analysis to a relaxation series, the first component, this is the first component spectrum on the left, that shows us the collection of all the signals that have about the same relaxation evolution in that pseudo dimension, whereas the second component shows us all of those peaks whose relaxation behavior is different than the majority. So this is a very quick way, without any kind of relaxation analysis, just looking at principal component analysis spectra, that we can see where the spectra are changing over the course of a series, and we can see it very quickly and easily. Now, in our lab, where we are interested in characterizing biopharmaceuticals to do things like, say, whether a newly processed lot is good or bad, we want to have quantitative ways to say whether or not two very similar spectra are statistically similar or not, according to whatever differentiation we're trying to make. Now, here's a case of two kinds of spectra overlaid, two monoclonal antibody spectra. Can you look at these and say which cases show spectra that are the same or spectra that are different? And I don't know about you, but even though I've looked at a lot of these spectra, I can't easily gauge that from a display like this. But I'm going to show you today that we can use principal component analysis, and in specific instances, we can actually use it in a quantitative way to make decisions like this one, where we can generate principal component analysis scatter plots and actually put confidence limits on them so that we can make a clear distinction. So for example, on the right, we have spectra that are measured from a new sample and a 30-day-old sample, and those samples are not really substantially different, and the PCA results can show us, yeah, these spectra are really spectra of the same material. Whereas in this case, I have two kinds of samples that I will talk about in more detail. They are samples that have been oxidized in two different ways. And while we can't easily distinguish those differences by a quick inspection of the spectrum, PCA reveals this and can actually be used to quantify this distance. So we're going to talk about that. Now, as we discussed, principal component analysis can be thought of as a kind of projection method. So let's think about a two-class case like this, where we have 5 replicate measurements of one class and 5 replicate measurements of another class. And we do our principal component analysis, and we make our principal component projection and look at the scatter plots. And we have what we can consider an interclass distance between these two clusters. So we would like to use this in a quantitative way, but the problem is, the challenge is that if we add, say, a third class of samples like this with another set of replicate measurements, when we calculate a new principal component direction, that component is going to be different than it was before, even though the underlying data is the same. The direction of the component is different, and so the direction of the projection is different. And so the features of the projection are largely the same, but the distances have been compressed or expanded. So the new interclass distance that you see when a third class is added is not the same as the old interclass distance that we saw when there were only 2 sample measurements. So in practice, if we want to quantify this kind of distance, we can really only do it quantitatively when we have only 2 sample classes. And so let's look at what that looks like in detail. And in fact, a better way for us to understand the behavior of this kind of quantitative analysis is to understand PCA as a matrix decomposition technique. Okay. And so in the example, we're going to use synthetic data with 2 sample classes. So both the samples have 2 peaks. One of the peaks is the same in both samples. The other peak is a little bit broader and in a slightly different position in one sample with respect to the other. And in this analysis, we're going to assume that the sample itself doesn't change over the course of the measurement. So there's no sample degradation. And the measurement doesn't change over the measurement. So there's no field drift or anything. And the difference between the measurements of a given sample are largely just random ones. So we'll have 5 replicates of sample A, 5 replicates of sample B. And if we looked at an overlay of those 2 kind of spectra, we can see the data that looks like this. And the whole series would look something like this, the way we see on the right-hand side. So this is input to principal component analysis. And in the principal component analysis procedure that we're going to use, we use an algorithm called NIPALS that extracts components one at a time. And to extract the first set of components, we use linear least squares procedure to find a set of scaling values and a synthetic spectrum that best describe the whole spectral series. So in other words, what we will do is make a linear combination of all the spectra in our input data. And that linear combination will generate a kind of synthetic spectrum. And the score values are the scaling factors that were used to generate this linear combination. And together, the score values and this component spectrum describe the overall series in the closest way possible in a least squares sense. So in other words, we decompose the series of spectrum into a set of score values and a synthetic spectrum. So we can multiply these score values by the synthetic spectrum and subtract them from the original data to generate a residual like we see on the right-hand side. And that residual is used as the input to the next calculation of the second component. And so in this case, we do the same thing. We have the collection of residual spectra, and we find a set of scaling coefficients and a synthetic spectrum that best describes this entire spectral series in a least squares sense. And if there are two samples with no substantial measurement variation or sample variation within each class, two component spectra should be sufficient to describe the entire series, regardless of how many replicate measurements it has. And the whole thing looks like this. So we have our input spectral data. We determine a set of scaling factors and a synthetic spectrum that best describe it. We multiply these two things together and subtract that from the data to generate a residual spectrum. And then we repeat that procedure. So we generate one component spectrum, which looks kind of like a sum of all the spectra in the series. And we generate a second component spectrum that looks more or less like a different spectra in the series. And the two of these are a good approximation for the entire data set. So we've compressed all of the data into two components. And if more than two are needed, it means that there was some kind of systematic variation, either in the sample during measurement or in the instrument across measurements. So this is also now diagnostic of whether there is intrasample variation or intrasample measurement variation. Very cool. So let's look at results for this specific example of a monoclonal antibody that was oxidized in two ways. And in particular, there's clinical importance for the pattern of oxidation of methionine residues on the surface of these antibodies. So we prepared two kinds of samples. One was oxidized in solution just by dumping in a bunch of peroxide. And a second sample was first put on a protein A column. Protein A binds to antibodies in a particular way. And so the protein A binding will tend to protect particular residues on the surface from oxidation. So one sample was oxidized in solution. The other was oxidized while bound to protein A, so that we expect a slightly different pattern of oxidation in the two cases. In practice, both of these samples are almost completely oxidized, almost completely. So if you actually look in the area where the methionine signals are supposed to show up, they're completely absent. And we have to reduce the contour levels by a factor of 5 to see anything. So the gray parts in this plot here show what the methionine signals usually look like. And in blue is the sample that was oxidized in solution. In red is the sample that was oxidized while it was bound to protein A. So there are a few more methionine signals, but they are very, very tiny. They're below what we would usually see when we inspect the spectrum. And so we want to see if we can use this PCA method to quantify the difference between these two kinds of samples. If we use just ordinary principal component analysis, we can see very good separation between the two kinds of oxidized samples. And if we look at the principal component analysis spectrum, we also see something very interesting. The parts in red and blue are the higher component spectra that show us where changes are occurring. And we don't just see changes in the methionine region, the changes in the spectra are throughout the spectrum. So there are interesting structural and chemical shift changes that are happening with respect to these two different kinds of methionine oxidation. So we want to see, how can we extract this information? How can we quantify it? Well, one thing is, if we do something simple, like just change the amount of random noise, we find that we can do this quantification effectively with this particular data, even when we increase the signal to noise content from a factor of 2 or 3 higher than it is to the original data. So that is kind of interesting. And then what we did is use many different kinds of data treatment and pre-processing to see which ones would make a difference in our ability to distinguish between these two kinds of samples. So on this vertical axis is the interclass distance, how different the two samples appear. And here are different things that we did to the data. For example, we apply binning to up to 10 points. It doesn't change the results. We apply smoothing up to 6 results. It doesn't change. We apply phase distortions, and this was kind of surprising. Even up to about 10 degrees, in this particular case, we can still separate data. But the thing that really kills the results is if there are systematic distortions in the data. So we introduce synthetic baseline distortions, and even at baseline distortions that have an RMS of about 0.2 or 0.3%, which is about half the random noise level, just these small, barely perceptible baseline distortions will destroy our ability to make this differentiation. And so the important take-home message here is that experimental technique is very important when you're going to employ principal component analysis in this way, because it's very sensitive to systematic distortions and artifacts. Now, here's another aspect that's kind of interesting and surprising. As we saw from the principal component analysis spectra, the differences in this case are distributed throughout the spectral data. They're not just in one location. So it turns out that regardless of which spectral region that we use as input for principal component analysis, we can still get good separation of the results. So that's another interesting thing about principal component analysis when we apply it to an entire spectrum at once. Often the information that we want is distributed throughout the spectrum, and so the results are robust with respect to which region we analyze. Now, here's another thing that's fun to keep in mind. It's a little bit surprising, but if you think about it for a minute, it makes sense. The information in time domain NMR data is the same as the information in frequency domain NMR data. And so when we present both of these kinds of principal component analysis data for principal component analysis, we actually get the same results. So this quantification works the same way when we apply it to spectra, as we see on the horizontal axis, or the corresponding interferograms, the way we see on the vertical axis. And interestingly also, this example is spectral data that's non-uniformly sampled. So if we do a regular reconstruction with a method like IST, we can get separation like this. But we can also, as an alternative, not even use any special non-uniform sampling reconstruction. We could just reconstruct this data with a regular Fourier transform instead, and the result looks like a mess to the eye, but the information that we need to make classification is still in here. So this is another interesting thing about using principal component analysis on the spectra. There are many cases where we could use a measurement technique that would not generate a spectrum that could be analyzed visually, but the spectrum still contains the information you need to make a useful classification. So in overview, what I hope I've convinced you of is that PCA is an excellent way to get an overview of any spectral series, and it has the special advantage that it's just as easy to use with 10 spectra as it is with 1,000 spectra. And as we've seen, we can use the PCA scatter plots in different ways to reveal different kinds of similarity and difference between classes of spectra. And we can use the PCA loading spectra to show us in a way that spectroscopists understand which parts of the data are changing over the course of a series. One important thing to keep in mind is that these methods use spectra and compare them directly on a point-by-point basis. So that means that issues of alignment and scaling have to be addressed carefully, and we'll hear more about that later. And we've seen that PCA can be robust to a little bit of extra random noise, but it still requires good measurement technique because you need to measure spectra that are as free from systematic distortions as possible. This includes baseline distortions and any distortions that might be introduced by the pulse sequence. So you want to use pulse sequences that are as artifact-free as possible. And then finally, we've seen that in a specific case, when we have two class cases, we can use PCA in a quantitative way to measure the differences between two kinds of samples in a quantitative way. So that is my overview. Here are some of the colleagues at NIST that we've done this work with, including our group boss, John Marino. And in fact, if you found any of this work interesting and you think you'd like to do some of it, tell folks that we have some very nice postdoctoral opportunities in our labs at NIST. So that is my overview. Thank you very much indeed. Would magnitude calculation be helpful? Okay, so that's a question from Istvan. And Istvan, actually, that is a great question. And we've actually done these experiments. And in many cases, we can get the same similar kinds of results with magnitude calculation data as we do with phase correction data. The caveat is that depending on what kind of classification you're trying to do, it may be that a magnitude result does not have sufficient resolution to make the kind of classification that you have in mind. Okay, we see another question. Using PCA, what pointer benchmark indicates when differences become significant? So that's excellent, and that has to be addressed on a case-by-case basis by characterizing the method the way that I just showed you. And the reason why it's different on a case-by-case basis is that in some cases you may be doing a classification that's based on a very subtle difference in a spectrum. And in other cases, you may be doing a classification that's based on an obvious difference in a spectrum. So all the cases have to be characterized for each kind of classification task. Any other questions? Would binning help against misalignment? Yes, it does. And as we've seen in the particular instance that I showed you, because we have zero filling and so on, and NUS doing extrapolation, we already know we have a larger number of points in the spectrum than we measured in the experimental time domain data. So we expect that we should be able to bin the data by a certain amount. In our case, we can bin the data by 8 or 10 points and still get the same kind of results. And as you point out, yes, that does reduce the difficulties introduced by spectral alignment. That said, you have to always check the situation about alignment. And then our final question, does the robustness of PCA allow lower field measurements? I'm going to defer that question to our next speakers. Okay, so who do we have next, actually? Right. So Professor Powers, next, of University of Nebraska.
So Frank provided a great overview of the utility and the power of using in terms of PCA. And PCA is a valuable tool, but it's kind of a means to an end. And what I'm going to focus on and discuss is some of the issues or problems or things to think about in terms of using PCA. And I'm going to do it in the context of metabolomics. So when we think about metabolomics, right, and applying PCA, the main goal here is giving our collection in terms of biological samples and whatever type of analytical data, NMR, and mass spectra we can use to kind of characterize the chemical profile of that. Can we fundamentally determine, are the metabolomes different between those different samples? And if they are, what are the major features or metabolites that are discriminating in the various different groups? And then can we go ahead and do a further network analysis? So conceptually, using PCA in this manner, it's a relatively simple idea. Take your samples, put it in the NMR, run it through a piece of software to generate your PCA model, and there you are. But the problem is if we just use things like that as a black box, we run into the classic problem of garbage in, garbage out. But since getting a PCA model, statistical model at the end is not the endpoint, but it's allowing us to go ahead and use that information to infer something about the overall biology of the system. If what's going in is biologically relevant, and even though we might get what appears to be a legitimate model, we might get actually wrong inference in terms of the significance in terms of the system. So again, if you think about preparing a biological sample and going ahead and collecting NMR mass spectra from that sample, you can get a wide range of sources of potential errors associated with that sample preparation. And all of these are going to lead to a problem in the resulting PCA model, right? And this is particularly true when we're looking at metabolomics, because metabolomics are particularly challenging for a number of reasons. The primary thing is that the metabolome itself is inherently unstable in terms of the processing and handling it. The application of protocols, including statistical techniques, are not uniformly established, and unfortunately, a lot of times are routinely applied incorrectly, which again, you might get what appears to be a good model, and the interpretation might seem valid, but it could be far off what the actual truth is. And of course, correctly identifying and detecting all the metabolites is a current major concern right now. So fundamental to using PCA or any statistical technique when you're looking at the model at the very end is the assumption that the metabolomes that you're comparing are biologically relevant. Any differences in terms of those samples are a result of the underlying biology, not to any of the steps in terms of sample collection or data processing. And I think this is where a lot of concerns come into play in terms of using PCA, right? So again, just to point out to people who are familiar with metabolome, kind of the obvious, again, the metabolome is very sensitive to handling, right? Metabolites can get converted into other compounds through enzymatic activity, and just simply sitting at the desk bench, the metabolites have various different levels in terms of stability. They can oxidize or get converted into a particular another metabolite. It's clearly something that we don't have to worry about when we're working with proteins or genes and so forth. You're not worrying about your protein suddenly converting into another protein by simply sitting on the bench. but with metabolites, that is going to be a seriously concern, right? So this is kind of some dirty laundry to illustrate the underlying problem in terms of just using PCA, any other type of statistical tool, as a black box, just simply applying it onto your data set and not really understanding what went into it. So here's a situation where a student was manipulating and processing a large number of samples and decided to go ahead and take a break between the morning to go get lunch and come back. And what we see is that the largest variation between these two data sets, not was the fact of the underlying treatment, was what samples were handled in the morning versus which samples were handled in the afternoon. So as Frank pointed out, the underlying approach of PCA and all of these techniques is to find out largest variance, regardless of what the nature of the source of that variance. Of course, we're all assuming that the variance is whatever our goal is, whatever, in this case of metabolomics, that goal is going to be, differences between biofluids collected from healthy patients or someone suffering from disease. And again, these techniques are incredibly sensitive. So here's, again, another real data set that was collected where two students were working on the same samples, following the same protocol, working side by side, but the major variance between those, which student handled what sample versus which student handled the other sample. And again, that was going to emphasize the major difference. Maybe in this case, it was like a junior student who was learning the process and not having as good pipetting skills as the first students. But again, the point is, at the end of the day, the model is telling you a group separation. It's onto us to determine what is the underlying meaning to that group separation. So again, we've been talking about PCA, and PCA falls into this class of statistical techniques, which are called unsupervised. And again, that's a really important aspect in terms of PCA, that it is always going to tell us the sort of the largest variance in terms of the data, regardless of the underlying nature of the data. The problem with the alternative techniques, like supervised techniques, where OPLS is one of those examples, is that we're actually telling the software the class membership. And again, in both cases, both techniques want to find a separation between a group, but a technique like OPLS supervised, it's going to try to force that separation. So this is not our results, but this is a publication a number of years ago. We're actually showing that if you put it in random noise data, you can still get nice group separation in something like PLS. And our results showed the same thing. So we started off with real data that had real good separation, really pretty separation. This is real. And then we purposely went ahead and add noise to the data. And PCA behaved as you expected. As more noise came in, the between group variance and within group variance started merging, and eventually we saw no separation. OPLS, though, because we're telling it to find group separation, it maintains that group separation. Scary, if you compare the features that are defining this group separation versus the spectral features here, there's literally no correlation. So if you were to interpret this data and identify metabolites associated with this, it would have absolutely nothing to do with the biology. So another concern or problem with the field in terms of interpreting the overall quality of your resulting model is uses of R-squared and Q-squared values. Now, these are valuable. They're informative. They tell us something about the underlying quality of our model. R-squared is like any type of traditional fit. It's telling you basically how well our individual data points fit the overall data model. And a value of 1 is going to be perfection. Q-squared kind of gives us a measure of the internal consistency. How well do individual data points fit the model when they're actually left out of the actual model calculation, right? The problem is that routinely R-squared and Q-squared values are used to inform the validity of the model, and they don't tell us that. And again, here's an example where we know this is an invalid model because we added so much noise and we got no group separation. But in the corresponding OPLSD model, it is again showing group separation. And the R-squared and Q-squared values of this model are actually fairly acceptable, especially the R-squared. But if you look at a validation statistic like the CV ANOVA, you can see that this model is completely invalid. And so validations of models are essential, using things like CV ANOVA or cross-validation permutation, not just R squared and Q squared. Another common question that comes out routinely is how many samples or how many replicates do I need per group to get a acceptable and reliable model? And the quick, simple analysis is the more the better, or as many as particularly practical. And the issue is that we're kind of playing up against this P versus N issue, right? Where normally we have a small number of replicates, but we have a very large number of variables, particularly when you go into in terms of mass spec. So as illustrated here, right, with a very, very simplistic statistical model, right, just simply assuming that we have 128 NMR bins and either that bin exists or not, if I get down to a small number of replicates, 4 or 5, the likelihood of at least one bin being consistently different between the group by just random chance is the probability of 1, right? And of course, with more variables, more signals, this issue becomes problematic. Fortunately, if you increase the number of replicates, this probability of finding this randomly a single correlation that has nothing biologically quickly diminishes. We can also see that in the group clustering separation that we were seeing, and when Frank was showing those nice scores plots. And again, if we look at a plot in terms of what the inherent group variance is versus the significance of group separation, I think that was one of the questions that we saw, is that we see again that the significance of the group separation as a function of the internal group variance again gets greater and greater with the number increasing replicates. And again, this kind of makes intuitive sense. So again, the more replicates, the better. That's going to, in terms of getting statistical significance and between your groups. So we've been talking about PCA, and like I said, PCA falls in this class in terms of unsupervised. And it's an extremely valuable tool. And as Frank alluded to, and I'm restating here, what PCA is going to do is find the largest variance in your data set, regardless of what your desired goal was. Again, I showed you those examples where the largest variance is due to the fact that the students, took a lunch break, right? And other common techniques are used are either PLS or OPLS, and both these fall into the supervised, which again can be very dangerous techniques if they're not properly validated, because both PLS and OPLS will almost always give you group separation. They're designed to give you that particular group separation, but they need to go ahead and get validated. The advantage in terms of OPLS is that by definition, that group separation is going to be forced into basically the first components, and everything else perpendicular to that are going to be confounding factors. And so there has some value there if I can go ahead and compare what we're going to be doing for PCA and OPLS. Personally, I am very against in terms of using PLS because, well, OPLS puts all your defined variance in one and all the confounding in the other. PLS still maintains it in both parameters. And that's why PLS kind of fits along the diagonal, while OPLS group separation is along the axis here. So one of the things that we can do or try to get out of in terms of a PCA after we see group separation is that we're going to want to understand what is the major features, peaks, or in the case of metabolomics, what are the metabolites that are associated with that group separation. But to be perfectly honest, it's a little difficult to do that relatively definitively because we have these multiple components and all the variables, as Frank explains, kind of contribute to the actual position in terms of along these components. So I might be able to go ahead and look at the general trend or a vector of a single bin or a single value in terms of how it might be contributing to movement in terms of one direction. But to make a clear contribution or make a biological insight, that could be a little bit problematic, which PC you're going to use. Like I said, with OPLS, it greatly simplifies that because it only puts it in one predictive component. And the result is that we can get these very pretty backscale loadings, these pseudo spectrum where it tells us the relative intensity and direction of the peaks that are associated with the group separation. And this becomes much easier to go ahead and interpret. Another thing that's essential in terms of with PCA or any of these techniques is that the data has to be normalized. Seeing any data that is not normalized, the meaning of that interpretation that it basically has no meaning. The idea in terms of normalization, we're catching whatever inherent variation that might be in the preparation of the sample, how many cells there might be, or how many, the volume of the relative concentration of their urine or blood sample is, right? But then one of the problems becomes, okay, which normalization technique do you actually use? Well, we went ahead and did a little exploration of that, and we found out that there's actually one really core technique. So I encourage everybody not to use histogram matching. That kind of failed very well. Most normalization techniques perform very well under modest stress. We go to extreme stress. When I'm talking about stress, I'm talking about large variations in terms of peaks intensity and large addition of noise. We see a couple techniques that kind of come out and perform best, which are listed here, right? But which technique you apply will give you a completely different model and then conceptually a different interpretation. So that's also something to be concerned of and aware of, right? What is going to be the emphasis of those normalization techniques? And again, in that analysis, what we saw is that most of these normalizations do very well in terms of handling dilution factor, right? Relative differences in the total concentration of your sample. We can go to extremely high and we almost get perfect reconstructions. The Achilles heel was noise, right? As soon as we got, even with the best performing normalization, as soon as we start to get about a 20% noise relative to our peak, that's when the methods fell off. And so Frank already alluded to this, and I'll re-stress this. Noise is our Achilles heel in terms of these statistical models. In fact, we learned this early on. As much as possible, you want to remove the noise from your spectrum. We were just learning about this. This is going back well over a decade ago. We just made some very simple samples, literally one ATP sample, 1 to ATP glucose, high concentration, collect replicates. You look at these, they're identical, essentially infinite signal to noise. We expected to see just like really tight clustering. When we get this model, we were really shocked and surprised and confused about it. What was the source of particularly the in-group variations? We went and looked at it. It was due to subtle noise variation in terms of the spectrum. I mean, really blowing it up. These little noise spikes was creating all this within group variation. So we went ahead and removed this. And again, we get an order of magnitude tighter group clustering. So again, noise is bad and we remove it all the time. Again, reiterating it, basic fundamental question, you know, what signal to noise do you need? Again, from the NMR perspective, we did a simple test, made a similar type of sample and collected 1 scan out to 32 scans. And you can see there's almost a linear relationship. As we improve our signal to noise, we improve our within group clustering and between group clustering. That's the only difference. The exact same set of samples, the exact same spectrometer, just simply running them with different signal to noise, and we see that. So remove the noise, improve your signal, as many replicates as possible, kind of the basic things. There was an answer to a question about using binning, and there's various different approaches to that. We prefer an adaptive, intelligent binning as opposed to uniform. We basically bin it around and identify particular peaks. We've always done this, and we've always wondered, okay, are we losing information by binning as opposed to using the full resolution spectrum with really good alignment? And to our surprise is, in the concept in terms of PCA, we are not. We get the exact identical result if we bin or we use the full spectrum. The downside is PCA is a relatively slow algorithm. If you use a full resolution, getting the exact same model takes you much longer. Conversely, when you're using something like OPLS, we don't bin, we use the full resolution spectrum, taking consideration in terms of making sure that we align our software choices, ICO shift, and the reason by using the full resolution, then we can get those pretty pseudo spectrum in terms of those backloading models to go ahead and analyze things. All right. There's been a lot of other techniques that have been coming out in addition to PCA. And so the question has always been, which works better? So we used a bunch of simulated NMR data sets with known variations. And the basic take-home message is, if you're dealing with generally good, robust data, none of the models really difference each other. If you really stress the data, very small group differentiation, a lot of noise, then OPLS was the actual method that came out better. The metabolome is complicated. We only observe a few hundred, and there's probably thousands, hundreds of thousands of metabolites. There's no reason why you have to restrict yourself to a single analytical technique. In fact, we use both NMR and mass spec. And this nicely summarizes how we get a broader coverage of the metabolome by combining both these techniques. Basically, there are metabolites detected by NMR that weren't detected by mass spec and vice versa, and there are some metabolites detected by both. Nicely, at least qualitatively, they had the same general overall trends. And there are modifications in terms of the PCA, particularly the multi-block PCA approach, that allows you to actually combine multiple data sets. Essentially, each Spectra goes into its own block. We get a sub-level, and then we kind of get the super block level, and we can get a full model of that. And the result is that by combining data, we can get much better models. And here is a real-life example where the NMR by itself, mass spec by itself, didn't really show very nice group separation. Combine these two data sets, and we got really pretty separation. And on top of that, you can still analyze each individual spectra, see the contributing features to that particular model in terms of this particular approach. So that's my quick run through. This is kind of a summary of some of the issues I kind of touch upon, and maybe some additional ones, specifically relating PCA in terms of metabolomics, things to do and not to do. Again, the metabolome is very sensitive, so handling is very impactful. Things to do in terms of using various different statistical techniques, what the information we can get out are going to be very different along those particular lines. And of course, this is my group. Most of the students doing the work here at UNL, and a lot of my collaborators on these particular projects. So with that being said, I'll go ahead and if there's any questions, or pass this on to our next speaker. So I see there are a number of comments in the chat. So there's a question about, could you use PLSDA to prove that a sample set is not statistically different? Does that make sense? I reviewed a paper that claimed it as a technique, and I couldn't judge the validity of that. Actually, I would argue that PCA is probably a better choice than PLSDA. As Frank said, and I've repeated, that if PCA is going to find the largest variance in the source of data. So if you generate a PCA model and it comes up like a scatter plots, there's probably a high likelihood that there's no statistical difference between those two samples, at least from a multivariate analysis. Whereas something like PLSDA, especially if it's not validated, could give you the false impression, because it almost always gives you group separation. And so it can give you the false impression that there really is a statistical difference between those two. So I would not use PLS, PDA, or OPLS as a means to determine it. So what we do in our group is always run PCA first, and if we get good group separation, then we go ahead and do the OPLS to identify what are the underlying variable differences between the actual model. And another question is, when you're using PCA on combined heterogeneous data, NMR and GCMS, how do we go about with normalization, scaling, transformation? So again, that's a good question, and generally that's done independently on each data set by itself. The main issue in terms of doing like a multi-block issue, which again, we use our software, NVA PAC software, so it's kind of built in, is actually scaling between the two data sets in regards to the number of variables, right? Since something like GCMS is going to have a lot more variables than NMR, if that relationship is not kind of normalized and scaled, then the GCMS data will overwhelm in terms of the NMR, in terms of identifying major features. So we have to kind of normalize basically the size of those data blocks. But then the standard normalization scaling is done independently with the NMR and GCMS data first. I think there's another question. Is explainability an important issue of saying diagnosis? Do we care? You know, that's a great question. I've seen people try to do that time and time again in the literature, simply trying to attribute some generic factor to any variation along PC1 and PCA or along PC2. And I think that's relatively dangerous to try to infer anything out of that. I think that type of diagnosis really is much better applying it to something like OPLS, when you know that in that one coordinate, everything contributed to that separation is due to your group definition. As soon as you get into PC1, PC2, and in turn to PCA, multiple variables are contributing to that variation. And of course, if you have multiple samples in there, it's, you know, what samples are contributing to what factor along that particular dimension. I think it's very difficult to deconvolute that. No, so there's a question here about, is it enough to just scale your data, e.g. Pareto scaling, and not normalize this? No, actually, no. You need to both normalize your data and scale your data, and they're having both two completely different goals, right? The normalization is accounting for, you know, just an intrinsic global variation in the quantity of your metabolites, right? You know, think about it along the lines of, say, you're collecting a blood serum, and in one case you get a half ml from a patient, in another case you get 1/4 ml. Then you lyophilize those down and dry them up and dissolve it up. Well, the larger volume that you started off with obviously is going to have twice, you know, approximately twice the total concentration. And that's what you're trying to adjust in terms of normalization. That dilution factor there is applying to. The scaling, what you're dealing with is the large dynamic range variation in terms of individual variables, right? You don't want a small change of a very intense variable to dominate your model, right? Again, imagine something that is, you know, on the altitude scale, the peak is at 100%, right? And then you have some other peak that might be only 1%, right? Well, 50% change in that peak would indicate a much larger difference in terms of that metabolite than, say, a 2 or 3% change in the most intense peak. But without scaling, the intense peaks are going to dominate you in terms of models. So you need to do both. Pareto scaling is one of our preferred methods that we use. All right. So I think those are right. So I'll pass this on to Emmanuel. So thank everybody.
Yep. Thank you, Bob. Okay, so in my part here, I will focus mostly on different applications that involve PCA using NMR data. And especially for metabolomics and targeted profiling studies, PCA is the most commonly used multivariate statistical analysis method. And it's really in the heart of all of these metabolomic studies. So there is a large number of very diverse applications of PCA with NMR. So here I will try to briefly show you some examples that I believe that they are characteristic, but have in mind that there are more than that. So again, when we have a project that involves spectral comparison, just by doing a visual inspection is almost always impossible to identify patterns among samples and make rigorous comparisons. For example, here we can see the NMR spectra of beer samples, and we're looking for differences between types of beers. Okay, it's really impossible to identify if there are differences between beer types and where these differences come from. In other words, which metabolites change among samples. Okay, this is why we need chemometrics and sometimes machine learning. And of course, PCA is the main technique we use. So Frank and Bob covered very nicely all of these things. Just a few things to remember about PCA. It's a class discovery technique. So we generally try to avoid the word classification with PCA. we say clustering, okay? Just in terms of wording. It's unsupervised clustering. And also it's not generally used as a statistical test. And you like when we have when we do a t-test or an ANOVA, we can say that there is a statistical significant difference between these two groups. In PCA, we generally say there is a good clustering or there's not a clear clustering among samples. PCA is strongly recommended for any project that involves chemometrics. Okay, this means that even if you don't have a good clustering among samples, it's important to provide in your publication or your report the PCA plot, because it's indicative of data, of the quality of the data. Okay, very often we work with systems that the variance is really high, so a PCA plot doesn't really show any clustering. And we need to do other things, like Bob mentioned, OPLSDA, or we can do random forest or other things, but we still need to show the PCA plot. Another thing to remember is that PCA it's used very often to identify outliers, means samples which are not really related to the rest of the population there. Very often people just remove the outlier. My recommendation is that go back to your, if you have outliers in your PCA plot, go back to your raw data, to your NMR data, see the spectra of the outliers. And if you don't see any technical issues like an issue with water suppression or a baseline distortion or a phase issue or whatever, then you may want to keep them there, okay? Because they may represent variance of future samples of the general population, okay? Also, as I said before, in systems with higher variance, like clinical studies, a clustering in PCA, it's not often achieved, okay? And that's absolutely fine. When the expected effect, let's say you have a control on the treated sample, but you believe that the effect of the treatment will be really very small compared to the total variance. In such cases, it's very important to try to increase the number of samples. Bob talked about that also, okay. Now, before we do PCA and other statistics, as Bob mentioned, there are some actions we need to take. So I don't really want to add more than this, but I just want to mention that the way you do the data pre-processing, means to prepare your data for PCA and other statistics, is really important, and it will determine the outcome of your analysis. So be careful how you do binding, normalization, spectral alignment. Also, a comment about transformation. We often need to do transformation like log transformation, for example, or fourth-root transformation, just to make the distribution of our data to look better. So when we do this, if you work with absolute concentrations, have in mind that after the transformation, your data may lose their quantitative nature, okay? So have this in mind. Also, if for some reason you cannot remove the noise and you may have zero values there or even negative values because there is also negative noise, then log transformation can be problematic. So you may need to consider another type of transformation. Now, after we do all of these things here, the output of all of these steps, it's something like that. Okay, so it's a table. In this table, we generally represent samples in rows, okay? And variables are chemical shifts, like it's an area, it's a signal area under a certain chemical shift as columns, okay? Still, it's a complex thing here, okay? So we did all of these things, but we haven't achieved really any dimension reduction. Our data is still very complex with many features and many samples. A visual inspection still does not reveal any difference among samples and groups, okay? This is where PCA comes and takes us from something like that no human being can deal with, okay? With something like that, it takes us to something like that anybody can understand. It's a 2D or a 3D plot, a scores plot in this case. And as we already learned today, we also have the loadings plot. So this is the dimensionality reduction, which is achieved by PCA. Now, the majority of applications of PCA in chemometrics are related to proton NMR. This makes sense, of course. NMR is a very easy and sensitive nuclei. There are also many databases and literature about the proton NMR assignments of different compounds and also on different matrices. So it makes sense that it represents the majority of the applications. So here you can see a typical application of PCA using proton NMR data. It's related to the authentication of avocado oil and its distinguishment from other edible oils. So this is the NMR spectrum, and this is the PCA plot, which is built based on this data. Okay, and you can see different oils cluster make different clusters in the PCA plot, and here you can even see that some samples that were labeled as avocado oils, actually they are soybean oils. So this is a typical application, okay, in food science. Now, again, we said that proton NMR represents the majority of applications, but you can also see literature applications with heteronuclei. For example, carbon. Not only carbon, you can see also phosphorus. People who deal with ATP and phospholipids, but carbon is also another application. Carbon, it's often ignored by people, but it can be a good alternative in some cases. For example, if the amount of sample is not an issue, or if you're not necessarily focusing on low concentration metabolites. So especially if you have, let's say, an observed probe, which is optimized for carbon, carbon NMR can be a good option. So here they used carbon NMR for coffee classification. The reason they had to do this is that in the proton NMR spectra of coffee, they had some issues in terms of chemical shift drifting, because if I remember correct from this paper, trigonelline and the chlorogenic acids, they were interacting, and this interaction was sample dependent. So they really had a hard time to, to align the spectra. But this type of misalignment did not appear in carbon NMR. So they used carbon NMR, and you can see the PCA scores plot. They were able to achieve a cluster. Now, in addition to 1D applications, you can see now in literature more and more 2D NMR applications, and Frank also showed a few. So now there are tools that you can treat 2D data. So you can do binding, you can do scaling, you can do all of these things in 2D NMR data. And this is a good option for people to consider, given the complexity of the 1D spectra, especially when you're working with samples of biological origin like plant extracts or foods or biospecimens. The 1D spectra can be really complex there, especially proton. It's really complex. So HSQC can be a good alternative. Here they used the HSQC, I'm talking about natural abundance, okay, to study yeast metabolism. And as you can see, different yeasts and different time points, the time course also. And you can see that the results between proton NMR based PCA, which is a traditional application, and the HSQC based PCA are comparable. I mean, which means that these applications here on HSQC works well. Of course, we can imagine that it's more time consuming, but on the other hand, it has some benefits. For example, you can find more loadings, more biomarkers using HSQC, because you can imagine in a 2D spectrum, you have two different frequency dimensions, so the dispersion of the peaks is much better. So they were able to achieve a better biomarker selection. Some years ago, you didn't see very often 2D based PCA, but now there are applications. Now, all of these are liquid NMR based applications, but given the high quality spectra that can be now generated by HRMAS probes, PCA and of course other chemometrics or machine learning techniques can be applied using this data. Here you can see an example where they used PCA to discriminate cheese based on sensory analysis. So there are different quality, sensory quality groups. And they did the PCA on HRMAS. So you can see, I assume that these are the big peaks from the lipids that appear in the cheese. But then you can see all the metabolites. It's a very high quality spectra, so why not applying PCA on it? Now, I'm talking so far about food-related applications because this is my field. However, this is not the only area that is using PCA. Of course, there are many applications in other fields, including health and disease. Here you can see that here they use it for breast cancer patients, and it's just an example of this paper. So you can see that there's a different clustering between control, healthy subjects, and let's say patients with breast cancer. Now, what we can see here is that, in my opinion, this is a good clustering given the data they work with. Okay, because in clinical studies, clustering can be really, really difficult in PCA, but that's not always the case. Here you can see that there is some clustering. Okay, just have in mind again, clinical studies have a lot of variance, and the factor you're looking for, it's probably not the main driver of the total variance in your system. Another promising area, in my opinion, that I like it a lot, is the coupling of PCA with data obtained by low-field instruments. Okay, low-field instruments are much cheaper, they're user-friendly, they fit in an industrial environment, but as we know, spectra suffer from overlapping, strong chain couplings, and all of these things. So PCA can deal with all of these problems, at least in some cases, and it is often able to extract information from this low-field data. At least in our lab, we haven't done much so far on this, but it's certainly an area that we would like to focus. Okay, so here they used PCA to classify precursors of fentanyl precursors, to classify the precursors of fentanyl precursors based on synthesis method. Okay, and they compared high field, 300 megahertz is still a high field, with the 43 megahertz, which is a low field. And you can see that they were still able to have a good clustering in the PCA plot. A nice visualization tool in PCA is the Biplot. And Bob showed one earlier. So Biplot simply overlaps the scores plot and the loadings plot and helps us to easily visualize the reasons behind any similarities or the similarities between samples and groups. Okay, so here you can see they have different honeys, different types of honeys, and you can see also the loadings. And again, what we said before, a loadings plot is not going to tell you, okay, this is, let's say, glucose, or this is trigonelline, or whatever. It will just give you a chemical shift. So at this point, let me try to stop sharing for a minute. And let's try to have a short demo here. So let me share again. Can you see now my screen again? Okay, great. So I have a small data set that we can use here to play. So this data set is made by Fen, who is also attending the workshop. Thank you, Fen. So it's 10 samples, 5 pomegranate juice, and 5 apple juice. Pomegranate is coded as 1, apple as 0. Okay, a very small data set. Each row is a sample, each column is a variable, a chemical shift. Okay, so we will use this, and we will use MetaboAnalyst, which is a web-based free tool for metabolomics. It's not the only one, of course. There are many others, excellent tools. I will just use this one. So before we go here, if you notice, there are even some negative values, okay, in some areas, not everywhere, okay, but here there are positive, of course. but there are also some negative values. Okay, so as Bob and Frank mentioned earlier, this comes from noise. Okay, so it's good to remove noise. If for any reason you don't remove it, you may have to deal with some difficulties when you do transformations, as we said before. So let's try to see how we can do a simple PCA analysis on this small data set. So again, each row is a sample, each column is a variable. So this is Metabolomics, okay? We click here to start. We choose, they have different things here, but we can choose statistical analysis. Now they have concentrations, spectral bins, or peak intensity tables. We will use spectral bins. We will choose a file. Let's put this one. This was made also for my metabolomics class. And we'll submit it. We don't want to edit the groups. We will proceed. Now, people sometimes do a type of filtering. Okay, filtering means, for example, remove the noise. Some other people, by filtering, mean, okay, I will remove all the variables which do not have a lot of variance. So there are many things, and this really depends on the person and the study. I will keep it very simple here, and I will put none. Okay, now it's where we do the normalization. I will, for sample normalization, I will put none because I have already normalized this data. So it's normalized by SAM, okay? Now, if you do not apply any normalization, and so this is how it looks, okay? So if you apply, for example, Pareto, and you normalize, now you can see that you have a better distribution, okay? If you want also to apply log transformation, you have also a nice distribution. I like Pareto mostly. Sometimes I have seen that auto-scaling works also very good. Depends on the data always. Okay, so let's do a Pareto, for example. So we can see now how this is before normalization, this is after normalization. Ideally, you want something like that. And then we proceed. So now we come here and you can do the statistics you want. You can do from a univariate thing like a t-test or some other things. We care about principal component analysis here. And you can go to your 2D plot and you can see the separation between pomegranate juice and apple juice. And then you can go, sometimes if you think that adding a third component, because now you have two components here, okay? Two principal components. If you think that an additional component will really have a significant variance, you can see the 3D plot, okay? Most of the time we show this one, and you can also have your loadings plot, and your loadings plot, of course, will just give you a chemical shift. It doesn't tell you if this is trigonelline or if this is formic acid or whatever, okay?
That was an excellent explanation and overview. Thanks very much to both of you. Everyone feel free to unmute your microphone and ask questions if you can. And otherwise, I will ask some questions and go back and maybe clarify some answers to questions that we already talked about. Okay, we have one question. Is there a difference in precision using eigendecomposition for PCA or singular value decomposition? Computation time is very different. Okay, unless either Bob or Emmanuel would like to answer this question, I guess I will.
I'm sorry, Frank, I haven't done that type of comparison. So
okay, so the question is whether an iterative method like NIPALS that calculates components one at a time is as good in terms of precision as singular value decomposition, which calculates all of the eigenvectors and eigenvalues simultaneously. So The challenge with that other method of using SVD is that you need enough memory to hold the entire collection of spectra in. So if you have a collection of spectra with several hundred spectra, solving the problem by SVD will often be prohibitive. What we've seen in examples with 1D and 2D NMR spectra, and by now we've accumulated hundreds and maybe thousands of examples, is that the results that we get from NIPAL's PCA are more or less identical to what we get from singular value decomposition PCA, with the one exception that depending on how data is normalized, we don't always get the components off in the same order as SVD has. And so we commonly reorganize the components from largest to lowest variance when we do computations with NIPALs.
I just wanted to remind everyone, this may be a good opportunity if you want to unmute yourself and ask directly questions. Don't have to go through the chat.
Right. Adam, you have a question.
Yeah, unmuted. I mean, that was pretty quick, Frank. Well, first, thanks to you and all the presenters. These are some really great presentations, some interesting topics to discuss. Pretty simple question, thing that's tripping me up a little bit. Some of the scatter plots, Frank, that you showed had principal component 2 versus 3, and some had principal component 1 versus 2. Why wouldn't we want to start with a 1 versus 2 plot and see largest deviation in data for clustering? Why would you go to the next level?
That's an excellent question. And part of that in the examples that I showed you has to do with the particular protocol that we use for our data, which is that we don't subtract the average before we start the principal component analysis calculation. We use uncentered data so that the first spectrum, the first component spectrum, is more or less like an average spectrum. And that first component does the job of centering the spectrum. So in many cases, the information that we want to see shows up in the next pair of components. It's a good question.
So I can kind of jump in with a little bit of a follow-up there, Adam. So especially with PCA, right? Because, you know, it's an unsupervised technique. It's going to be looking at and finding the largest variance, and that largest variance is going to be put in PC1, right? And so, you know, if you have some insider information, which again can be dangerous, but if you can attribute, as exactly as Frank was saying, you know, some other non-interesting factor that's really dominating PC1, then you can maybe argue that, okay, what I'm really looking for, whatever my biologically relevant or significant question I'm trying to answer, maybe that's simply moved down to the second, third tier in terms of the PCs, right? But if you don't know that, and so I have seen people do this, and I've seen this in publications where, you know, someone's looking for a difference, or looking for what might be the best appearance in terms of the actual scores plot, and they might just randomly pick a pair of PCs that kind of give them the best clustering, but it's simply based on, well, this gives me the base looking clustering, as opposed to, well, I have some knowledge or insight or some other explanation in terms of why I chose, you know, PC3 versus PC7 or something like that. So it can be very dangerous if you just simply randomly pick things.
Thanks. That makes sense. Although it seems like it could be then just dangerous by randomly picking, because if this is a coordinate system change, reacting everything onto these principal components, presumably there's going to be some principal component find that might show you what you want to show. Yeah,
exactly. You know, that unintended bias. And of course, as you go lower in terms of the number of PCs, the importance of the contribution of that PC to the overall model drops off pretty rapidly. And you also run into the problem, which again would be comparable in terms of PCA. Are you putting out too many components there and simply overfitting the model, right? Take a a trivial, simple example. If you have this normally XY data and you fit it to a straight line, it's like, oh, look, my R squared is only 0.6. I'm not really happy to that. Let me go ahead to an n-fold polynomial fit, and I'll get my curve to go through exactly every data point, and I'll get an R square of 1. Look, brilliant data. You can make that same argument in terms of PCA, right? Right.
Excellent. And there is another question in the chat, which is where you declare the number of components. So just to be clear, when you use SVD, you have to compute all the components. It always computes all the components, whether you want them or not. When you use a method like NIPALS, you can decide ahead of time how many components to calculate. And generally, with NMR data, which is relatively sparse, all the information that you want is in a relatively small number of components. And if you have 2 sample classes, 3 components should show you everything.
Yeah, I agree. We very rarely with PCA do we go, go beyond 2, and occasionally go to 3. And usually going to 3 is if it's a complex data set with multiple different in terms of groups. So yeah,
okay.
So you either choose it manually, or some software will actually go ahead and do a analysis and determine the actual statistical significance of additional PCs that are being added into it.
Right. Okay, Rachel, I see you've turned on your camera. Do you have a question?
Yeah, I have a question about the case when you have for each sample measured NMR spectrum, and then some result of another experimental technique which gives just one-dimensional information, just number, and how to deal with the fact that from NMR spectrum you get thousands or hundreds of dimensions, and then from another experimental technique you get just one dimension to make sure that it will be somehow actually counted in the results.
Can you explain the question again? I'm not sure that I caught the details.
I'm dealing with the problem that I have the data set of hundreds of samples, and for each of them I have NMR spectrum, and then there are results of another experimental technique which gave just a number, but it's some chemical activity.
Oh,
and
so you're talking about the case of combining different types of data. So I'll let Emmanuel and Bob answer these questions because they work with multimodal data. So that's what you're asking about.
So Rachel, I'll apologize. At least on my end, you're breaking up again. So I'm going to repeat to make sure I understand your question was. So you had, say, 100 replicates where you have an NMR spectra for each of those replicates, right? But then in addition to that, you have maybe a single value measurements, let's say like a an assay result or maybe a clinical evaluation. Is that, and you want to combine those two? Is that your question?
Yeah, exactly.
Right? Yeah. So I can envision multiple ways to go about doing that, but in all those approaches, it kind of falls into a classic supervised approach, right? So PCA would only work in kind of like an afterthought, right? You would apply your PCA model, like, exactly like Emmanuel and we all were showing you, to your NMR data sets, and then you get your corresponding membership in terms of clustering, right? And you can maybe simplistically, color on or label onto that figure your individual values to see if anything comes up. That's a little tedious and that's a little manual, and that could be problematic. Alternative, you can take like a technique like PLS or like OPLS, where normally when you just run that generically with not having the information you have, and you're normally doing that with just simply 2 groups, control versus treated, whatever that happens to be, the y-axis that's in that OPLS model, we just arbitrarily set those to 0 or 1, right? Because group 0 and group 1. But in your case, what you would do, you'd actually apply your number, whatever that number to that y-axis, right? And that would then drive the OPLS model, right, to see whether or not the groups are going to separate out or form clusters based on the value of that particular, whatever that measurement you actually have. And so we've done that. We've done that numerous times, and sometimes that can work out and be informative, and sometimes not, depending on how well the NMR data correlates with that measurement that you actually have. But again, if you do that, you have to be very careful and make sure, like I was talking about, you have to make sure you properly validate that results, okay? Because the OPLS will give you force groups based on that number, right? And then you have to determine whether or not that's actually valid. But that's how I would do it. I think that would be the first pass. Apply OPLS, use your values as the y-axis to go ahead and define, basically define the group based on those numbers.
Okay. Thank you.
Yeah.
Okay, I'm going to jump in. Yes. So I am interested in more of the way that PCA can be used for ligand binding studies. And I was curious, if we were to analyze the binding of a mutant protein, if principal component analysis would be, I guess, affected because of the mutant itself rather than the difference in binding of that ligand.
Right. So that's a great question, and it depends on how you organize the data for PCA, right? So if you included everything, you'd have some spectra that are native protein and some spectra that are the mutant. And even without ligands, those are two different sample classes. And so when you add ligands, you would multiply the number of sample classes. That said, it's possible that looking at all the data might reveal particular cases where both the native and mutant spectra have spectral features in the same place that react to binding in the same way. Okay, so it's a completely case-by-case basis. So my guess is that to use PCA effectively on that data, you would have to analyze the native type data separately from the mutant data. But totally easy to do a third analysis where you include all of them together and see what's what. And just that analysis will tell you which parts of the spectra are common between those two cases, and you can potentially dismiss them for certain kinds of analysis. So that's my comment. I don't know if Bob, who has done this kind of work previously, has some comments.
Yeah, so again, I completely agree with everything that Frank just said there. And clearly the simplest way, the quick way would be treating them as four separate groups, right? You have the mutant and the native with and without the particular ligand. But if you want to try to get into, fine details in terms of trying to get differences or similarities between the native and mutants, you probably would have to do some pre-processing. And so the way that I would think about it is, presuming the mutant's a single amino acid, and you know what the assignments are for the two. And so you, instead of having, as Emmanuel is actually showing, that's two-dimensional matrix, right, of bins, I would think about it would turn into a two-dimensional matrix of amino acid and the delta chemical shift that you would actually measure, right? So Frank, I think your software probably would be able to do that very quickly in terms of measuring those delta chemical shifts across a relatively large value. And then those deltas could kind of go into the PCA analysis. Right.
And so that's actually, so in other words, there are two very different approaches that would be interesting to do this way. One is PCA on the chemical shifts, and the other, and not on the chemical shifts, but on the chemical shift differences.
Right, yeah.
And then on the spectra itself. Doing it on the chemical shift differences is very interesting for your particular case, right? Because let's say in one case, there's an alanine in the protein binding pocket, and another, there's a glycine. So we might expect that if, say, an aromatic group sticks on top of those two peaks would move about the same amount. They would have the same kind of chemical shift perturbation. So by analyzing the chemical shifts directly, you might be able to eliminate some of the variation between the native type protein and the mutant.
Awesome. Thank you both for your feedback.
Yeah,
Appreciate
great
it.
question. Thank you. Anything else? All right, this was okay.
I just have a question.
Yeah.
So, yeah, Frank, you showed that figure where you had the minimum interclass distances of the PCAs.
Yeah.
And the varying binning and line broadening and all these things. I was just wondering if anyone has made something similar, but with magnetic field strength, or if there's any kind of correlation between that.
So that's a good question, and I think we actually saw examples in Emmanuel's talk.
I saw you showed one, Emmanuel, right? The one with the 40 megahertz and the 300 or something. Yes. I was just wondering if there is some specific correlation, like if you double your magnetic field strength, for you, will you double the distance or something, or is it very sample specific?
So that was not that systematic to try to find correlations, according to the magnetic field change. But what we can see from this study was that the clustering was a little bit better in the high field. That's it, without making, but that's a good thing to, to consider, because, to find any correlation between the magnetic field strength and, quantify the changes in the PCA plot. They haven't done something like that.
Okay.
So it was only a pairwise comparison between the 300 megahertz and the 43, if I remember correct. That's it.
Right. The differences, though, are you can attribute some of the differences just to resolution. And depending on what kind of classification you need, there may be some critical level of resolution that if you drop below it, you can no longer make the distinguishing
so if you have some samples that are quite similar, then you probably need a higher field to get the good separation.
Right.
Yes, and one more comment there. So you're talking, of course, about the scores plot, but if you want to think a little bit further, it's also in the loadings plot. This is where you're going to have more difficulties, actually.
Right.
Because the low field, it seems that it looks like the IR-based metabolomics. So you can have a good separation in the scores plot, but then if you try to find biomarkers, it may become a little bit difficult.
Right. Now, what I think about this is kind of in the opposite way, which is that, as we've seen in the examples, there are many cases where PCA can pull out a significant difference, even when it is not visually apparent. So I actually think PCA will prove to be a very good way to exploit low resolution NMR data that might not be sufficient to assign a molecule, but will be sufficient to classify or characterize it.
Yeah. And combining low field with 2D, so you can run a series of HSQCs and you can have a better resolution there. This may be another way to approach it.
Right.
Okay, so there's a future for low field PCA.
Oh, absolutely. Yeah, no question about it. Good question. Thank you. All right. Any other brave folks want to pitch in with a question? Kenny.
How's it going, Frank? Great
talk. To hear from you. What's up?
All right. Have you, by any chance, tried running PCA on crafted antibody spectra?
I have not, but Krish has. Unfortunately, he ditched early.
Something tells me the combination of the two has a lot of potential.
Absolutely, because you can selectively filter components in the craft table. You can remove all the small ones. You can remove the ones that you don't have assignments for, or you could just keep the ones that you don't have assignments for. There are any number of useful things that you can do. And of course, you can do the analysis either on the table or on the spectra. So there's a huge number of things that you can do with the craft data. And Krish is the best one to, to give an outline of that. So we'll ask him next time.
Thank you.
Anything else?
So, do people have more questions?
All right, I think this was splendid. We did have lots of nice questions, and we had tremendous presentations from Emmanuel and from Bob, and it was a pleasure to participate. Thank you all very much. John and Dave, do you have any concluding remarks?
I'll turn things over to John.
You know, once again, you guys just keep raising the bar here for us, and therefore we will certainly have to continue these meetings. And as I said when we opened up, we certainly plan on doing this for quite some time to come. And thank you everyone for joining us today. To our wonderful speakers, very interesting topic matter today. And all I can say is watch your email inboxes for invitations to get more upcoming meetings. And thank you everyone. And Dave, I'll turn it over to you once again for any final remarks.
Okay, I have no more final remarks. And so thanks everyone for being here, and I'll close the meeting.
Thanks, folks.
Thank you very much.
Bye. Thanks, everybody.
Bye.